I guess we should already start. Our session now is about testing anti-aging treatments on humans, the good practices, the good stuff. So it is my pleasure to start this session with the presentation of Dr. Barzilai, who everybody knows here, who will speak about TAME, targeting aging with metformin, the news about it. Thank you, Nir. Welcome.
Thank you, Ilya. Thank you, everyone. Let me make actually some provocations that are more related to the subject of animals versus humans, and I think I'm going to make very important points. And please realize, TAME is an after day work for me. Okay, that's not my science. I had grants and merit award for animal studies. I'm a geneticist and have this longevity study. So I'm working between models and animals. And, I have this study where we have centenarians. We have actually almost 750 centenarians and their offspring, and we're looking for longevity genes. And the first thing that we wanted to know really is all this data from animals on longevity, does it make sense from what we know about the genetics of centenarians? On the genetic of centenarians, there are two public genes that always appear in centenarians. It's the ApoE2 and the FOXO3A. The FOXO3A certainly is related to mechanism of aging in animals. We have genes that are specific for humans. For example, CETP is not a gene that is expressed in animal at all, okay? So we have to expect that animals are conserved, a lot of mechanism we learned there, but there are also going to be genes that are centenarian specific. I would just say that if there are 7 billion people in the world and we would have the whole genome sequencing and electronic medical record of every one of them, we would probably be able to cure every disease in the world. And that's what pharmaceuticals are doing now. They're leaving those animal models that have been disappointing for specific diseases, and they're looking for genetic proof. And in fact, our CETP and ApoC3 discovery have led to drug development by Merck and Ionis. So this is what we did to answer the question, are the mechanism in human and animals are the same? We have whole exome sequencing of our centenarians and also of people who are not related to them. And we looked at genotypes that were less than 1% of the population, rare genotypes, because centenarians are rare. And with Zhengdong Zhang, who's a computational system biologist, geroscientist, the first thing we did is actually see which are the variants that are showing up more. But the most important thing is we have to put in those variants into pathways, because we are not made out of one variant at a time. We are made out of many variants that can influence the pathways upstream or downstream. They can amplify, they can be instead, they can cancel, depending what there is. And by checking a phenotype base that comes later, we, with this unbiased approach, discover really something really interesting. The top pathways are the mTOR, that's for longevity. Okay, that's what's different between centenarians and control. mTOR signaling, insulin IGF signaling, MAP kinase signaling. Okay, so the message here, this provocation is to tell you, yes, what we learned so far is very relevant to human longevity. We should breathe freely. We should use this information that will be published soon to say, yeah, our animal models are good. Now, what do we do with this information? Well, we can also go back to animals, and we can go to animals and not backwards. In other words, not discover things in animals, but go backward when we discover in humans. So we found a mutation in the IGF growth hormone signaling pathway, and you all know that animal models, when they have impaired function of IGF-1 of growth hormone, they live longer. In fact, in our centenarians, the women with the lowest IGF-1 levels lived almost twice as long as the women without the IGF level. And by the way, 60% of our centenarians have some genotype on the growth hormone IGF axis. So what do we do about it? Well, Amgen has actually created an IGF receptor antibody, which we borrowed and murrinized and gave it to animals. And you see here that those animals that were old, 22 months old, extended their lifespan by 10%. Okay, so animals are not lost. It's just we have to use them in both direction. And by the way, their health span was fantastically improved. Their cardiac function, their exercise capacity, their sensory motor function. And remember, it's generally not too late. There is probably too late for some things to do, maybe not for senolytics, but basically it's not too late to treat elderly. Okay, so next point. The reason we have TAME is because FDA doesn't have indication for something that is like aging. And we have always a discussion if aging is disease or not. My point is always it's not necessary. But if we don't have a recognition from the FDA, if aging is not preventable, then healthcare providers are not going to pay for our drugs and pharmaceuticals that we need. We need to move from biotech to pharmaceutical. They have to take our drugs to phase 3 trial. They will not develop, and they want that better drug, or like we heard before, a combination of drugs to really have an impact. So metformin, we recently showed evidence that it targets all the hallmarks of aging. And so I want to correct something that we heard before about metformin is only diabetes. In fact, metformin is not really so relevant to diabetes, but to the fact that it's used for diabetes. It's been used 70 years, but guess what? It was used to prevent flu and malaria. And at that point, it was discovered that it lowers glucose in diabetes, so it's now diabetes. But metformin is not primarily a diabetes drug. I also should tell you there are 9 studies now across the world that shows that metformin lowers mortality of COVID patients by 50 to 70%. Not 7%, not 10%, but more than that. It's generic, it's cheap, and we do TAME to show that it targets aging, but more important, we do TAME. Metformin is a tool to give us an FDA indication. I should just say one thing. The data that we saw before of effectiveness of drug is from animals, okay? So if you look at data, and I'm not showing you here, if you take every data on metformin that shows its effect on aging, the effect is about 7% in animals. In humans, metformin decreased mortality in diabetic patients 17% more than non-diabetic, and 50% more than diabetic, okay? So the measure of longevity in animals is skewed a little bit. If metformin have a lot of effect on immunity and inflammation, we're not going to find it in our labs and in our experiments, okay? So let's not take all this and pretend that we know what the effects are, and let's be prepared to the fact that there are things that will target aging, and they're not going to be effective in mice for a variety of reasons. So metformin, the nice thing about metformin that we have, the TAME study has been done, okay? But it has been done on separate disease. Metformin prevents diabetes, metformin compared to insulin and other drugs prevent cardiovascular disease. There's association studies, hundreds, that people on metformin have less all kinds of cancers, maybe except prostate. Metformin prevent cognitive decline in clinical studies and in Alzheimer association study, and I already talked about the mortality. So we're talking about a genetic chip safe that is a tool for us to do TAME. But this is the point. This is what we have to do, biologists. What we've been doing is doing studies where we take drugs. I'll show you the metformin, but we did this with resveratrol, we did it with acarbose, we did it with exercise, we did it with exercise and metformin, basically taking old people and giving them either a placebo or drug, taking biopsies of adipose tissue, of muscle, doing a washout, and switching. Those that were on treatment are now on placebo, those were on placebo are now on treatment, and looking at the biology, okay? The clinical part is okay. It's improving, glucose and height glucose and insulin under the curve and insulin secretion. But this is the data that I want to show. Again, underlying the mistake that you think that metformin is a metabolic drug. Yes, pyruvate metabolism is expected if it has metabolic effect. But the transcripts that are coming here are of DNA-based excision mismatch, of NAD biosynthesis that has things beyond metabolic, BRCA DNA damage, and in adipose collagen chain, remodeling, and LXRX that has also genetic influence. The point here is we're talking about a drug that extends its action below metabolic. You can see here in pink and blue in muscle and adipose the metabolic effect, but the point below here is really important because we look at the upstream regulator of those transcripts. And guess what? Again, mTOR comes up, MYC comes up, TNF-alpha, TGF-beta. Okay, again, we get changes in the transcripts that are in humans, those transcripts that we convinced ourselves that are important in aging. My final slide is just to put up this study that we hope to start as soon as COVID allows us, where we take 3,000 people, and what we're looking for is to shift the time until we get any one of those incidents of disease, cardiovascular, dementia, cancer, and death. This is what the FDA wants to show. Ilya, thank you. I'll stop here, and if there's any question I can answer, if I have time.
Yes, we have 3 full minutes, so we can have 2 or 3 questions. So please.
Could I ask a question?
Yes.
Harold. First, 2 questions. One question is, have you tried applying Horvath's clock to any of these animals after metformin treatment?
Well, the answer is no. First of all, the clock for animals is a little bit more complicated. I don't want to get into that. What I'm trying to do, there was this DPP trial, the diabetes prevention trial, where people who were 50 years old got metformin for 3.5 years on average. Some got it to 5 years. This is stored at the NIH, and we want to get access to the blood samples in order to do the clocks on those patients. Metformin prevented diabetes by 30%.
Okay. Other questions? Other questions?
Questions.
I'll read a question from the chat, and then Greg. Dr. Barzilai, you recently stated that the philanthropist has been found that will fund TAME and also start a huge longevity fund. Could you give an update, please? And also just curious also about the general update on TAME.
Well, look, we are ready to go. We feel lucky that we didn't start last year because it would be devastating to stop this study. Although, you know, if we started 2 years ago, we would probably have been showing that we prevent death, okay, in people who are on metformin. But the study is going to be funded through FR by, we hope so, The money is not in the bank, but there is a commitment from an organization that will announce itself, I think, next month. And I don't think I'm available to give more than that now.
Okay, please, Gregory Payne, the question. Unmute yourself, yes.
Good. Nir, does the TAME study propose to use one dose of metformin or different doses? What are they?
Yes, it's going to be one dose, and you know, it was the regular discussion. Well, we should give more because we want more effect. And some people said, "Are you crazy? It's elderly. You should give less." The reason we chose this dose is because all the studies that I showed you, just titled them, that's pretty much the average dose that they used. And it was important for us to have, and certainly for the FDA, to have some idea of what dose we had. If we had all the money in the world, we would have done a dose response, right? And see how it happens. Look, part of the problem with metformin in animals, and I hope you don't take wrong the way I say it, but metformin that targets complex 1 in the mitochondria is like a weak cyanide, right? So there can be too much of that. There's actually literature that's saying that if you do 10 and 100 times the dose, animal will die younger. So what's the sensitivity in animals? I don't exactly know, but because it's important for us not to kill anyone on the road to the FDA, that's the dose we decided, and we wouldn't know which is the best dose. And by the way, we wouldn't say what is the right age to start, right? Because we are taking people 65 to 80, and I would guess that, you know, the diabetes prevention was on 50 years old. So we think you could start taking metformin earlier than that.
So what was the dose, Nir?
1,500 milligrams.
Oh.
Merck for Germany is providing that. Metformin is very cheap, but the placebo is actually quite expensive. So Merck is kind enough to provide the dose.
Okay, thank you very much, Nir. That's the time.